picture of a chick embryo, and most of the initial studies of somatogenesis were studied in the chick embryo because it's so easily manipulated. Okay, so the somites are basically blocks of cells that give rise to muscle and skeleton, and they're shown here. You see these individual kind of squares that are colored in. And the somites will give rise to muscle and skeleton, and they are surrounded by what is called paraxial mesoderm. So paraxial just means on both sides of the axis. Okay, so you have mesoderm on both sides. And as you see here in the diagram, there is some mesoderm that is labeled the lateral plate mesoderm. So this is a specific mesodermal location that is formed during somatogenesis. Okay, so here's the head, and here's the posterior. Here are some somites that are forming. Okay, so if we compare the chick embryo at 24 hours, which is the picture I just showed you, and at 33 hours, you can see that there's more somites formed, right? So there's more blocks here. And you can see that if you compare this embryo, so here is some unsegmented mesoderm that has not yet developed into somites. And this is called pre-somitic mesoderm. So the pre-somitic mesoderm will develop into somites after that region called Henson's node passes it, passes that location. Okay, so pre-somitic mesoderm is the region between the last formed somite and the regressing node. Okay, so pre-somitic mesoderm would be between, say, this stage and this stage would be the tissue right in front of, or more posterior to Henson's node. Okay. And the regions can develop into 4 or 5 individual somite blocks, and they develop simultaneously on either side of what's called the notochord. So you basically get pairs of somites formed on each side of the notochord after Henson's node passes that location. Okay, so what's really interesting about the somitic mesoderm, pre-somitic mesoderm, is that even though it hasn't developed yet, even when it is not a somite, before it develops into somites, it already has a positional identity. That means that if you do experiments where you take a region of mesoderm that will eventually develop into vertebrae that is more conducive of the thoracic vertebrae. So let me take a step back. So are you guys familiar with the bones in your back, right? So you have cervical vertebrae, and then you have the more lumbar vertebrae, right? And so there's different kinds of structures of each bone depending on the location, if it's more cervical towards the lumbar region. And then around your lungs, you have another set of vertebrae that connects to your ribs. Okay, so there's different patterns of vertebrae depending on the location in your body. Same happens in the chick. And they're derived from pre-somitic mesoderm. So the experiment that was done was transplantation experiments where pre-somitic mesoderm that is in a location that will eventually develop into the more thoracic vertebrae, so the ones that look like more around the rib cage, were transplanted further up, so more anterior, into a less developed chick embryo. And then that embryo was allowed to mature. And what was observed is that in the in the area where this pre-somitic mesoderm was transplanted, the vertebrae on that side resembled more of its original faded features. So you were getting thoracic vertebrae up at the neck region on one side, but on the other side that had the endogenous natural mesoderm, you only got formation of just the regular cervical vertebrae. Okay, so this shows that even though this embryo has not yet developed this somite, it already knows that it's supposed to become a thoracic vertebrae at this stage, and that its fate is not plastic. It's determined, because when you transplanted it into a different region, it still became what it's originally thought it was supposed to be. So this shows that at some very early stage of mesoderm induction and patterning in the chick embryo, these somites already know or already assigned a location and a location genetic program. Okay, so the position of the somites along the anterior-posterior axis helps determine their fate. Okay, the anterior somites form the cervical vertebrae. Okay, so here's another interesting tidbit about the mesoderm. So the somites are produced at specific times and in a very specific order. And the order of somite formation is kind of pre-programmed as well in the pre-Semitic mesoderm. Okay, so if you do another kind of interesting experiment where you take the pre-Semitic mesoderm from an embryo, and so if this region X represents the more anterior end and Y represents the more posterior end, and you just invert it and then do a surgery where you transplant this inverted Semitic mesoderm in this chick embryo, what you get is an embryo that basically has that section of its vertebrae upside down. So there is a certain stage where you cannot force this mesoderm to, or you can't induce it to reset itself. So does everyone understand this experiment? Okay, so rearranging the pre-Semitic mesoderm even before they're formed doesn't change the timing. So what you see also is that even though you've inverted this tissue, after somite 5 is formed, then in this new transplanted embryo, somite 6 will form first before 10. Okay, so there's some specific timing as well as location that is dictating the fate of these somites. And the pattern is laid down earlier by some anterior-posterior axis signals. Okay, so there's a signaling pathway that is known to be activated during semitogenesis at very predictable times. And this is the Notch signaling pathway. So there's a target gene in the Notch signaling pathway called just C-HARY1. And it's expressed in the somites shown here in gray at very specific times. So this circle is just giving you a point of reference. It's showing you, I think it's showing you where in the embryo the node may be. And so what you see is that the C-Hairy expression kind of changes after each block of somite is formed. And